but I just have 2 PowerPoints to get through. It's a continuation of our introduction to the 4 major plant groups that we started last time. So this should look familiar. We introduced the 4 major plant groups, and then we spent more time talking about the bryophytes and then pterophytes and lycophytes. So maybe we can review a little bit about that today, and then the new stuff, we've got 2 PowerPoints, one about gymnosperms and then the other about angiosperms. You can see an additional detail down here about angiosperms that we added. There's going to be a distinction between 2 types of angiosperms. We're going to distinguish between monocots and dicots. Both of those are types of angiosperms, but we'll see differences between those. That's all that I have on this chart, but there is the lecture handout that you have about the gymnosperms, and I have another one that I'll pass out about angiosperms whenever we get rolling. Any questions about where we left off last time? We got all the way through bryophytes and then the pterophytes and lycophytes. That stuff, as well as gymnosperms and angiosperms, is all relevant information for lecture quiz 5, which is up on D2L, and the post-lab quiz is now up on D2L. It has to do with the introduction to plant types that we saw last time. And if you happen to miss that, I did bring them back on the side of the room, so we can go through and tour those things again. Sorry.
Thank you.
Yeah, no problem. And you were here last time, right? So you got the handouts about mosses and ferns. Then the only new handout so far that we've gotten to is this handout about gymnosperms. No problem. So we'll pick up there, make our way through the gymnosperms PowerPoint, through the angiosperms PowerPoint, and then I have a video that I'm going to show of angiosperms in action. And it's going to be a little weird because I'm messing with this computer and I'm used to being over here. So I'll just get used to that, hopefully. But stop me at any point if there are any review questions about our previous plant groups that we've talked about. Just as a reference, again, we've talked about bryophytes. These were our only example of non-vascular plants. Everything else had vascular tissue. So when we looked over here on the side of the room, the mosses, the ones that lack vascular tissue, are the only ones that couldn't grow very tall. These you can't easily see from all the way across the room, whereas anything that does have vascular tissue, like these ferns or these lycophytes, this we said was a club moss, not a true moss, because a true moss over here doesn't have any vascular tissue at all. It's hard to see with the naked eye. But when you have vascular tissue, it allows these plants to grow taller into the air and also further down into the ground to reach for water and minerals. Gymnosperms and angiosperms all have vascular tissue, but we said gymnosperms and angiosperms have something that the pterophytes and lycophytes don't, and that is that they produce seeds. Gymnosperms, that term just translates into naked seeds. So these seeds are just going to be exposed to the outside environment. They sit on these exposed female cones. And then the other picture that I have, I guess I lost my pointer, and I can't use that mouse, so I'll have to go over here. The pine cone that I brought in. This is an example of a gymnosperm that we're going to call the Conifers. That was going to be our main example of these gymnosperms. In our PowerPoint, we're going to introduce 3 other types of gymnosperms. Again, Conifer is going to be our main example. But there is another picture that I have right up here. This is another type of gymnosperm. It's technically a group that we're going to call the Cycads. The technical name for this is a Sago Palm. It's the female version of a Sago Palm, and those red structures that you can see there, those are the exposed seeds. So again, naked seeds just means that these seeds are exposed to the environment. If we think about what's inside of a seed, we're going to see this by the end of the PowerPoint, but if we could look inside that seed coat, there would be a baby plant inside there with a little bit of a food reserve, a little nutrient packet that's going to help get it started on its growth. So everything in terms of gymnosperms and angiosperms are going to have seeds. We're going to see that angiosperms take it a step further and are going to encase those seeds in a fruit. The fruit's role is going to help disperse those seeds. We'll see that angiosperms also have flowers, and the flowers is going to help attract pollinators. We'll get to those in a second. Again, stop me if I'm going too quick on any of this stuff. First PowerPoint is all about gymnosperms. Here's where we introduced 4 types of gymnosperms. The ones that we see in lab are the conifers, but there are 3 other types. Cycads are maybe one of the other common types that you might have seen. They are found in apartment complexes or in neighborhoods. They're planted. This version of a cycad is called a Sago Palm, and they come in male varieties and then female varieties. This is the female structure that produces these seeds. It's called a strobolus, and this is a male cone that's going to be releasing pollen grains. That's my hand right there, so you can kind of get an idea of how big those seeds are compared to the size of your hand. They're rather large for seeds, and that cone that you see on the male plant is about 3 feet tall. I used to have one of those, and I don't know where it's at. I will try and find another one to bring in here that we'll have as an example. So cycads are an example of these gymnosperms. They have naked seeds, which qualifies them as a gymnosperm. Not all gymnosperms are going to have cones like the one that we saw in lab, but to make it a gymnosperm, it has to have these naked seeds. So cycads are one example. Ginkgos are a second example of a cycad. So ginkgos, this used to be a more diverse plant group. It's an ancient plant group, and there's only one surviving species left of ginkgos. It's this ginkgo biloba tree. It comes in, there's a male tree, and the other one is a female tree. It's only the female tree that has the exposed seeds on it. So this is, it could grow here. Sometimes people will plant these as ornamentals and you'll see them. The characteristic leaf type is it has this fan-shaped leaf. You have a sketch of some gymnosperms in the handout that you just received from me. Some of those you should be able to recognize as this ginkgo biloba leaf. So we have cycads, we have ginkgo biloba. The third group of gymnosperms are going to be called Neetophytes. The example in the United States of Neetophytes is this ephedra plant. It grows in the deserts of West Texas. It could be, a common name for this is Mormon tea, because you could actually boil that down and make tea out of it and get ephedra from it. That's where this comes from, this ephedra plant. So it has naked seeds as well. We don't typically see this in our lab. This Welwitschia is another version of the Neetophytes, but it is found in the deserts of Africa, not North America. But the common gymnosperm that we see, the one that we've already been introduced to, are the conifers. It's the 4th group of gymnosperms and the most common. When we think about gymnosperms, we're going to again let conifers be our example, our common example of gymnosperms. I sometimes describe these gymnosperms as the reptiles of the plant world. There's a lot of common themes that gymnosperms have with reptiles, and follow me here because I'll say the same things with reptiles. What we see gymnosperms do that reptiles also do is they figure out how to break this link with water. Their reproductive life cycle is no longer tied to water. The previous 2 plant groups, you might remember from last lecture, we said in order for sperm and egg to meet for bryophytes, a drop of water has to be present. The sperm have little flagella on them and they have to swim over to the egg. The same is true for all of the ferns, horsetails, and club mosses. These all require a drop of water to be present for fertilization to occur. Otherwise, sperm and egg are never going to meet. They don't have to. Again, these have to stay in a moist environment constantly. Pterophytes don't. They're kind of like the amphibians of the plant world where they can move into a dry environment, but it's going to have to be wet at some point if fertilization is going to occur. So their life cycle depends on water to be present at some point. Gymnosperms and angiosperms are going to break that link with water. And the cool thing about that, if we think about the last thing that we talked about in 1406, we said when things happen, with either plants or animals where they gain access to new territory, it starts to separate these groups of organisms into different areas and they have to adapt to different conditions. Cold, dry environments or hot, wet environments, it leads to these different selective pressures. And when when it's either plants or animals that are gaining access to new territories, it leads to these bursts of evolution. We call those adaptive radiations. You see it in reptiles just like you see it in these gymnosperms. When they break their link with water and move to the interior of the continents, they really start to speciate in a wide variety of ways. So that's why there's so many different types of reptiles, and that's also why there's so many different types of gymnosperms. If we think about conifers as our example, they're going to include the oldest living plants. That's this bristlecone pine, one plant, over 4,000 years old. Its location is kept a secret so nobody damages it. It's somewhere in California. It also, this group of conifers also includes the largest living plants. That would be these giant sequoias. So these are like 2,000 to 3,000 years old. Not the oldest, but they're the tallest. I'll jump back to this as well. They're the most widespread. Again, they break that link with water and are able to move into territories previously unoccupied by plants. How they do this is that their reproductive structure is going to be cones. And cones, we're going to see, are going to use wind as their pollination method. We saw that video last time of those cedar trees in the hill country that were erupting, and it looked like just clouds of smoke that were moving through there. What was really erupting, we said it were these tiny male cones on the tips of these cedar trees. Those are packed full of pollen, and wind dispersal is not very specific. It's very wasteful. So if wind is going to be your method of pollinization, you just need to produce vast quantities of pollen and hope the wind is blowing the right direction that day so that some of those pollen grains are going to land on the female cones. Conifers use cones as their reproductive method. But I had mentioned last time about the confusion that I had about cones. I'll just pull this example of a pine tree that I brought in here. One of these branches we can see has a bunch of male cones on them. They're tiny. And this other one is kind of the woody cone that we typically think of. We need to be aware of these as female cones, and the smaller ones that are going to produce pollen, those are the male cones. So we have two different types of cones. We'll set those down for now. The other thing that we mentioned in our long discussion of this alternation of generation last time is that the first two plant groups, we saw that the bryophytes, it was a case where most of the time, most of the life cycle was spent in the gametophyte part of the life cycle. Only some of the life cycle was spent in the sporophyte part of the life cycle. I realize I'm saying these terms gametophyte and sporophyte, and it might be worth throwing those out there as a review question. So when you hear somebody say sporophyte, or let's start with the first one. Let's start with gametophyte. Let's say that cash cab pulls up in the parking lot. You're walking out of class, you're going to your car. Cash cab pulls up, is trying to give you $1,000 if you can explain what a gametophyte is. We wrote it down on the board last time. There's two things that really are descriptive of what a gametophyte is. I mean, you can describe what it does as well, but how would you describe a gametophyte? What is a gametophyte? I'll pull up a picture real quick because we're just going to review gametophyte and sporophyte as we go through these four major plant groups. And there I am at the wrong. Give me a second to figure out how to work this laptop. So we saw this slide last time. This one. And this was the slide in which we talked about this alternation of generation. In plants, they do something weird where there is a multicellular haploid phase. That's not something that we have. Our haploid phase, where we make gametes, they stay individual haploid cells until fertilization occurs, and then we get our first diploid cell that we called a zygote. So we defined gametophyte and sporophyte, I guess, with our visual picture here of a gametophyte. What were those two things that we wrote on the board that describe a gametophyte? I might have just slipped up and said them right there if you caught that. What makes a gametophyte a gametophyte? I'll get a sip of coffee while we get some answers formulated. I heard something. I heard one of those terms in there, and it was haploid. So the term haploid does apply to gamete. Sorry, let me try that again. Haploid does apply to gametophyte. It also applies to gamete too. Ethan, what do you have? Those are the two things. The thing that makes a gametophyte is that it is multicellular and haploid. That's right. Because there are some other things in here that are haploid, like spores are haploid and gametes are haploid, but they're single haploid cells, where gametophyte is a multicellular haploid structure. Okay, that's great. And then I think you said the word gamete, so it is also worth remembering that the gametophyte, this multicellular haploid structure, it makes gametes. That's where it gets its name. But how it makes those gametes is kind of unique. It makes those gametes, so I can ask this another way. What's the difference between a gamete and a spore? They're both single haploid cells.
Gametes are made from mitosis and spores are made from meiosis.
That's right. If you caught that, gametes in the plant life cycle, gametes, sperm and egg, are made via mitosis. The gametophyte is already haploid, so we want to maintain the chromosome number. So gametes are single haploid cells made by mitosis. Spores are single haploid cells made by meiosis. And the thing to remember there is this word sporophyte. The sporophyte makes the spores. So that's kind of easy to remember. Gametophyte makes gametes. Sporophyte makes spores. We have to remember the sporophyte is, what are those two things that describe a sporophyte? Kind of the same as, similar to gametophyte, but not the same?
Diploid?
Those are the two things, multicellular and diploid. So the sporophyte is multicellular and diploid, and some of those diploid cells are going to undergo meiosis and become haploid spores. Okay, so this alternation of generation in plants is that there is the gametophyte that produces a sporophyte, and then the sporophyte produces the gametophyte. They take turns producing one another. They alternate between this multicellular haploid phase and this multicellular diploid phase. Okay, good. Back over here. Okay, so right back to this one. Now we can remember in our little comparison of the four major plant groups, the mosses are the ones whose gametophyte part of the life cycle is the dominant part of the life cycle. It's the only part that is green and photosynthetic. It's the only part that has root-like structures that can pull in water and minerals. So the gametophyte dominates the life cycle, and it's the sporophyte that is dependent on the gametophyte. You can see the sporophyte doesn't have any green photosynthetic qualities. It can't make its own food. It doesn't have any root-like structures to pull in water. So it's totally dependent on the gametophyte for all of its nutrients. But then when we got to the ferns, we said the ferns, the gametophyte part of the fern life cycle and the sporophyte part of the fern life cycle are independent of one another in terms of nutrition. They both have green photosynthetic cells that can make sugar. They have little root-like structures that can reach for water and minerals. The reason that we said the sporophyte, we said the sporophyte is the most conspicuous part of the life cycle just because it grows large enough that you can see it with the naked eye. That gametophyte down there that is the little heart-shaped looking structure, that's about the size of a dime. So whenever I was holding up our fern plant over here, all of this dried out now, but all of this green fern fronds that I'm touching, this is all of the sporophyte part of the life cycle. So it's the most conspicuous part. Somewhere down there in the soil is a little dime-shaped structure that's the fern gametophyte. So even though they're independent of one another for nutrients, we say the sporophyte is the dominant part of the life cycle because it's the only part we can easily see. But after that, everything that we're going to talk about today, the gymnosperms and the angiosperms, you're going to see that it's the sporophyte part of the life cycle that dominates. And the gametophyte part of the life cycle is extremely reduced. One of the reasons that I wanted to say that word gametophyte again, now when you hear gametophyte, you're thinking more than one cell, and each of those cells are haploid. Look at how small those gametophytes are. We're going to see in gymnosperms, these male gametophytes are only like 3 haploid cells big. So they're about as reduced as you can get. and they're only show up seasonally. Like we said, those male pollen grains aren't as prominent throughout the year, but seasonally they show up whenever the plant's trying to reproduce. So the sporophytes are present year-round, the gametophytes are present seasonally. Let me see. Okay. So once again, comparing the four major plant groups in terms of their life cycle trends. The next thing that we mention in terms of gymnosperms is talking about the seed itself. Having vascular tissue is something that gymnosperms do, but vascular tissue isn't new. Pterophytes and lycophytes had that vascular tissue. What's new about the gymnosperms, we said, was the presence of this seed. The other key advantage that these gymnosperms have is that, again, they have broken that link with water. So everything prior to gymnosperms is going to need a drop of water for fertilization to occur. But by breaking that link with water, these things can, like we said, move into places that were previously unoccupied by plants. I'll say this a couple of times too. I had mentioned that when we get to gymnosperms and angiosperms, these plants are going to be producing two different types of spores. So our blue note that we put on the board last time was that they're on this line of plants developed this second characteristic that we called heterosphere. That these plants, they produce two different types of spores. There's going to be a male spore and a female spore. Male spores are called microspores. Female spores are going to be called megaspores. And I'm saying that again just because I want you to connect microspore with pollen grain. We see this term pollen for the first time. Pollen is blown through the air. It can cause us allergic fits. But what a pollen grain really is it's going to be the male gametophyte. It's a microspore, a single haploid cell that's going to divide into 3 haploid cells, and that's going to be the male gametophyte. It's small enough that it can be blown by the wind. Sometimes I will say pollen grain, and sometimes I will say microspore. Those are the same thing. They're describing what becomes the male gametophyte. And here's the life cycle of this conifer as our example. I know it's just a sketch of it here, but you can see on this branch of a conifer, like the one that I brought in for our lab example, there are female cones, and then there are these much smaller male cones. If we follow just the male cone, for example, there are these little scales, and inside each of those scales, the cones are part of the sporophyte. So all of those cells that make up the male cone are diploid cells. And a few of those cells inside the scales, a few of those diploid cells are going to undergo meiosis and become haploid spores. Now that we're talking about gymnosperms, we can't just say spore, we have to say either megaspore or microspore. These male cones make microspores. Microspores and pollen grains are the same thing. So these microspores or pollen grains are going to be blown by the wind, and what they're trying to land on is they're trying to land on the female cone. If we look at the female cone, it also has scales, and at the base of each of those female cone scales, there sits a diploid cell. Again, the female cone is part of the sporophyte. So all of those cells that make up the female cone are diploid cells, and one of those diploid cells, it's called the ovule at the base of that scale, is the diploid cell that undergoes meiosis. It becomes a haploid spore. We have to use the term megaspore because that describes the female spore that's produced. This megaspore, like any spore, is a single haploid cell made by meiosis. This megaspore is going to divide into a handful of haploid cells. The female gametophyte, I know this is a small picture, we'll see a bigger picture in just a second, but there's more than one haploid cell right there. There's about 5 haploid cells that make up this female gametophyte. One of those haploid cells is the egg. We see not that we see it in great detail here, but whenever the pollen grain lands on the female cone, fertilization is going to occur. There is a diploid cell that is formed, and that diploid cell quickly starts to divide and become a handful of diploid cells. That's what we refer to as the embryo, a collection of diploid cells. this embryo is growing and dividing, it's fueled by the food reserves that are basically the leftover cells from the female gametophyte. Only one of those cells gets fertilized and becomes the zygote and develops into the embryo. The rest of those cells of the female gametophyte are going to be the food reserves for that growing plant. So that's what's inside a seed, a little baby plant with a food packet to get it started on its life. For these cones, we're going to see the seeds are mostly going to be distributed by the wind. And I'll try and get a video, or I'll try and bring some examples of this in. If we can find some seeds, maybe on the scales of that female cone, if you've seen these fall out of a female cone, or if you found one on the ground, you can throw them up in the air, and they do this little slow helicopter descent. And that's what helps keep them in the air longer and carry them greater distances away from their parent plant. But wherever this thing lands, it's already got a little jump start on life. When the little baby plant pops out of the seed coat, it already has a little bit of a root structure going, and it has a few embryonic seed leaves that get it started on its growth and development. So we have our sporophyte that's going to grow and produce more either female or male cones. Let's look at those male or female cones a little bit more closely. This is the female cone that we talked about, and you can see those scales. It's at the base of each of those scales that we find this ovule. That's a diploid cell that's going to undergo meiosis and develop into the megaspore. Megaspores grow into the female gametophyte. Microspores grow into male gametophytes. There is the male cones. Those male cones are producing microspores. If we take a cross-section through one of those male cones, there is a cross-section through one of the scales. You can see it's just packed full of pollen grains, or microspores. Microspores and pollen grains are the same thing. When these pollen grains or microspores are released, they're tiny. They're easily blown by the wind. So pollen grain and male gametophyte are synonymous. That structure is the male gametophyte. It's 3 haploid cells long, or 3 haploid cells big. This is the video that we saw last time that shows the smoke coming, what looks like smoke coming from cedar trees, and it's really just those male cones that are releasing their vast quantities of pollen grains. That computer's not hooked up to the internet, so I can't show that video right now. We'll just have to remember that from last time. Or you can click on this link if you have internet, and you might be able to see that. What is really blowing in the wind? It's not smoke, it's these little pollen grains. And if we look more closely at one of those pollen grains, there it is. It's basically 3 haploid cells. There are 2 haploid, starts off as 2 haploid cells. And then one of those haploid cells goes through a stage of mitosis, and we end up with 3 haploid cells. You can see the nuclei of the 2 sperm cells, and then the 3rd cell is what forms this pollen tube. So just 3 haploid cells are what make up this male gametophyte. There is the male gametophyte with its little pollen tube. It's trying to land on one of these female scales. And at the base of one of those female scales, there's a little opening that's going to allow this pollen grain to enter and find that female gametophyte. If we see this process a little bit more closely, let's look at the female cone pictures quickly. Female cones are also called ovulate cones because they have the ovules at their base, and it's in those ovules that the megaspores are formed. Here is that scale. There is the ovule. That's the cell that goes through meiosis and forms our megaspore. There it is. There's the megaspore. Actually, let me take a step back. Nope, I was right. Just looking at the wrong part of this. That structure right there is our megaspore. We can see it's a single haploid cell produced by meiosis. It's surrounded by these cells, the leftover cells of the ovule. This was a diploid cell that underwent meiosis, and we have our first haploid cell. So we know spores are single haploid cells that are going to end up growing through mitosis to form this female gametophyte. So let's see the next slide. That megaspore, like I just said, is going to grow into the female gametophyte. It's going to make a handful of haploid cells, and only one of those haploid cells is going to get fertilized by the sperm. If we zoom into this a little bit, there's the larger picture of that. But this structure that we're looking at is representing the female gametophyte. It's more than one haploid cell. There is this large haploid cell that we can kind of see in the background, and then in the foreground we've got the haploid cell that is representing the egg. The pollen grain, when it lands on this female cone, is going to elongate that pollen tube that's going to deliver one of those sperm nuclei to the egg. It makes its way through a little opening in the female cone called a micropyle. So the pollen grain now fertilizes the egg, and what results from that fertilization is this embryo. There's a zygote that forms, that first diploid cell, and that zygote continues to divide by mitosis until it's a handful of diploid cells. We have a growing little baby sporophyte there. It's using the nutrients, the leftover tissue from the female gametophyte. Those are all haploid cells that now have become a food reserve for this growing plant. It's protected in that little seed coat as it grows and uses those food reserves. At some point it's going to grow large enough that it's going to burst out of the seed coat. Before it does that, we're going to see that the job of the seeds are to disperse this baby plant to a new location. Gymnosperms have a harder time doing that than angiosperms because it's much easier when you have fruits to pay off an animal or something to take the seed to a new location. Without fruits, a lot of these gymnosperms use wind as their dispersal mechanism. Nothing there. Here again is this life cycle of the pine tree where we can see those seeds have wings on them. They're going to help carry them further off away from the tree that produced them. So that was our introduction to gymnosperms. We have one more PowerPoint that's going to introduce some angiosperms, so let me pull that up real quick. I should have answered in my run-through of the conifer discussion all of those questions from your handout about conifers. If I missed one, let me know. I'll be glad to repeat. But I'm jumping to angiosperms real quick. We know angiosperms as the flowering plants. They also have fruits. So fruits and flowers are something that if you see, you immediately know you're talking about an angiosperm. So this is our last group. And again, we took this a little bit further on the board than we see currently on the screen. We can further divide angiosperms into what we're going to call monocots and dicots over there. So let's start with angiosperms. They're called flowering plants, so we see a flower. There's even a little pollinator that's on that flower. We'll talk about the roles of the fruit and the flower, and we might as well go ahead and do that now. So we're seeing a flower on the screen. The role of the flower in the life cycle of the angiosperm is to attract a pollinator. And this is a much more effective way of pollen distribution. Instead of just producing vast quantities of pollen and hoping that the winds blow in the right direction that day like the gymnosperms do, angiosperms can employ the help of a pollinator like a bee, or there's mammal pollinators, there's reptile pollinators, there's all kinds of pollinators. Insects are probably the most common. Any pollinator is going to be able to pick up a packet of pollen from one specific flower, from one location in the forest, and then by hunting down another flower just like it, is going to end up dropping that pollen off at a specific location somewhere else in the forest. So there's no waste of the pollen in between these two locations. It can be very effective in terms of how you can get pollen from one location to the next. It also spurs a lot of this coevolution between plants and their specific pollinators, as we'll see in a video. So the role of flowers is attracting the pollinator. The role of the fruit is dispersal of those seeds. The fruit itself is just this ovary wall that surrounds seeds. So we're going to run through some examples of what a fruit is in a second. When you go to the grocery store, anything that has an ovary wall that's encasing seeds is technically a fruit. So tomatoes or bell peppers, those are all fruits technically because they have an ovary wall that encases those seeds to the inside. And the role of this fruit, like we said, is to aid in the dispersal of those seeds. So we've got the role of the flowers and the role of the fruits out of the way. Let's look more closely at those flowers themselves. We've got one example of a flower in the back that's kind of wilty. So I'm going to bring some fresh ones in next week, and we're going to want to be able to compare what we're going to call perfect flowers from imperfect flowers. A perfect flower is going to have both male and female reproductive structures on them. Male and female reproductive structures on these flowers, they get called special names. We know flowers are on the sporophyte part of the life cycle, so the part of the flower that makes the female spores, female spores would be megaspores. That's the megasporophyll. Megasporophylls make megaspores. Megaspores make the female gametophyte. Microsporophylls make the microspores. So this is the male reproductive parts of the flower. Microspores, we know, grow into the male gametophyte. This is a perfect flower. We can see it has both female reproductive structures on it, the stigma that is sticky and it catches pollen grains. There's this long style, and then there's the ovary down here that houses those cells that we called ovules. Those ovules are the diploid cells. They start out as diploid cells, but those are the diploid cells that undergo meiosis and end up as haploid megaspores. Those haploid megaspores just hang out down there. They grow into the female gametophyte, and then they wait to be fertilized by a pollen grain. Pollen grains have to land on that stigma and then grow a really long pollen tube that goes all the way down into that ovary and finds one of those ovules to fertilize. Here's the male parts of the plant. The anther is what contains all of those microspores, and then it's found on the end of this stalk that we call the filament. In addition to male and female parts of the flower, there's the petals, which are the pretty showy parts of the flower that really attract the pollinators. And then you have the sepals, which are the still green photosynthetic parts to the flower. Sepals, petals, then male and female reproductive parts. If all of those are present, then we've got a perfect flower. If we are missing either male or female reproductive parts, if a flower like this one over here just has female reproductive parts, it's going to be considered an imperfect flower. This one over here, the closer one to me, has both female and those male reproductive parts, so we would consider it a perfect flower. These ended up being too fuzzy to maybe be helpful, but this colorful picture is showing a flower that has both male and also female reproductive parts. So it's a perfect flower. The other two, these are flowers that are either just containing male reproductive structures or just containing female reproductive structures. So these are imperfect flowers. It might not be immediately obvious to you. You might be thinking these perfect flowers have both male and female reproductive parts right next to one another. How is it that they prevent themselves from self-pollinating? They have ways to prevent it. I just threw that out there for you to think about it for a second. We'll answer it by the end of this PowerPoint. It's obvious. One easy way to do that is just to have female and male parts on completely different flowers, which is kind of how the juniper trees do it. This is a male branch that just has male cones on it. I had brought an example last time of a branch that had the little blue berries on it. That was the female version of the tree that just had the female reproductive parts. Okay, here's our life cycle of the angiosperms, and it is also, there's a lot going on here. We're going to zoom into parts of it, but I'll start up here with the flower. We know the flower has both the male parts, that anther and the filament. If we could just zoom into that anther, we said those are where you find the diploid cells that undergo meiosis to form microspores. We know the fact that it's a spore tells us it's a single haploid cell that was produced by meiosis. Again, sporophylls, the microsporophylls make microspores. That microspore is going to grow into a handful of haploid cells, really just a haploid pollen tube and a couple of haploid sperm. So 3 haploid cells basically make up this male gametophyte. It's very small, it's blown by the wind. It's also called a pollen grain. So it's trying to land on that sticky stigma found on the female structure of the plant. It has this long pollen tube that it's going to grow down to where it finally reaches the ovary, and it's trying to find one of those ovules to fertilize. We said inside that ovule is the only diploid cell that forms a female gametophyte. If we go back up to the flower and look at the female reproductive parts, we can see there is the ovary, and if we zoom into that ovary, there are a handful of ovules. Those ovules were the diploid cells that go through meiosis to form megaspores. Megaspores are a single haploid cell. There's the megaspore right there. It's going to grow into a handful of haploid cells. It's like 5 haploid cells. Those haploid cells all together are what make up the female gametophyte. Again, we see that word gametophyte, we're thinking multiple haploid cells. There's not very many haploid cells, but there's more than one. One of those haploid cells is going to end up being the egg that gets fertilized by the sperm, and all of the other haploid cells are going to end up forming just the nutrient-rich packet for the growing plant. That nutrient-rich packet we call the endosperm. And if we can zoom into this fertilization process, in angiosperms, we said this last time, we just name dropped it and didn't define it very well. We said there's something that happens in angiosperms that's called double fertilization. This just refers to the fact that 2 sperm nuclei are deposited to the female gametophyte. Here's where it starts. We can see the pollen grain has landed on that stigma. The pollen tube has grown down that style and is about to reach the ovary. What's making its way down that pollen tube are 2 sperm nuclei. We're going to see that one of those sperm nuclei fertilizes the egg, and that's where we get our diploid zygote. So 2 haploid nuclei fuse to form our diploid zygote. All of those other haploid cells that were the body of the female gametophyte are also going to receive one of those other sperm nuclei. And those collection of cells end up forming the endosperm in angiosperms. The endosperm in angiosperms is triploid. It has 3 sets of nuclei. It's a more nutritious endosperm than the haploid endosperm that we saw when we were looking at the gymnosperms. So double fertilization in angiosperms refers to the fact that 2 nuclei are deposited to the female gametophyte. One fertilizes the egg, making it a diploid zygote. The other nuclei is going to contribute to this endosperm, making it triploid nuclei. So a more nutritious endosperm. There is this seed coat. Inside is the developing plant, that embryonic sporophyte that is growing into a full-fledged sporophyte. It's using that endosperm as its fuel. And then we have that diploid seed coat protecting that young plant from the outside environment. In fact, I'm just going to jump to this one picture right somewhere in here. Promise it's up here. There it is. This is the picture of just one seed. I like it because it's so zoomed in. We can see a very thin seed coat, and you can see what's inside is basically a baby plant. It's a baby sporophyte. This is one that's almost completely eaten through its fuel reserves. There's very little endosperm left. What we can see, that little point right there, that's the tip of the shoot, the part of the plant that's going to grow up into the sky. This down here is the tip of the root that's going to go down. So before it even breaks out of the seed coat, this is a baby little plant that has a starter root, and it even has these 2 starter leaves that you see. There's that little line that separates the leaves from one another right down the center. So I just wanted to show this. We'll go back to the slide that I just left off from. This one. I just wanted to show that close-up picture of a seed. It's got a baby plant that's growing inside, feeding on that endosperm. At some point, it's going to run out of endosperm, and it's going to be grown large enough at that point that it's going to be ready to burst out of that seed coat. It's already got its little starter roots, it's got its starter leaves, and it's ready to grow into a sporophyte. Angiosperm life cycles. There are some sample pictures of angiosperms. We see the flower, so we know immediately we're looking at an angiosperm. This is an example of a perfect flower because we have female reproductive parts as well as these male reproductive parts. This would be another perfect flower. We can see these anthers that are covered in those pollen grains that they're trying to release. And in the middle there is the sticky stigma, there's the long style, and somewhere down at the base would be the ovary. So another perfect flower. Here is yet another perfect flower. That little structure right in the center, that's the stigma, the style, and down there is the ovary. It's surrounded by these anthers. You can count them. There should be 6 anthers. And we can see the showy petals here, these purple petals. Those are in numbers of 3. Back here, these are flowers that have 4 petals. Keep that in mind. We're about to use that as a clue. When we think about these flowers, their job really is to attract pollinators. There's a wide variety of pollinators, and one of the things that I find interesting is that these flowers, they can give off wavelengths of light that our eyes cannot perceive. The picture that you see over here is taken with an infrared lens that is able to pick up these wavelengths of light, these signals that flowers give off that our eyes can't pick up, but things like these bats or even bee eyes can see wavelengths of light that we can't. And they basically direct those pollinators right down to the flower. We've got a video that's going to show us a bunch of these animal pollinators. Insects are probably the most common, but we're going to see mammals, birds, reptiles, wide variety. Now, not all angiosperms use animals as their pollinator mechanism. Some still use wind as their pollination method. Grasses do a lot of that. But there's all kind of different varieties that flowers take. They can be extremely tricky. Like this is a dead horse arum. It's about 2 or 3 feet in diameter. So this is a huge flower. And if we could be close enough to it, we would quickly pick up that it smells like rotting flesh. It smells like a dead carcass. And its job is to mimic rotting flesh. Its pollinator is blowflies, these flies that think they're going to get a meal. And they fly into this thing. It even produces hair that, well, it's not real hair. It's these little plant structures that look like hair that's trying to mimic a dead body. So it tricks these flies into thinking they're going to get a meal when really they're just doing the flower's work for it. It's kind of crazy. So flowers attract pollinators. Once pollination has occurred and fertilization happens, then the flowers fall off. It's all about developing the fruit at that point that's going to disperse the seeds. If this was a botany class, we would spend more time separating the different types of fruits from one another. I'll just show a quick comparison of different types of fruits here. What makes a fruit? What a fruit is, like we said, is the ovary that surrounds the seeds themselves. So this bean pod, a green bean, is considered a fruit because it's an ovary wall that surrounds seeds. An apple is obviously a fruit. We can see this nice fleshy ovary wall that provides a lot of sugary nutrients as a payoff for any animal that's going to disperse these seeds to a new location. What's crazy is that all that sugar that the plant has pumped into this apple doesn't go at all into helping the seeds. Remember, the baby plant is already feeding on its endosperm inside that seed coat. So this additional nutrients that's been supplied to the outside of the seeds doesn't help the baby plants grow. It just helps get them to a new location. This is fascinating symbiotic work that happens when angiosperms figure out how to trick other animals, whether it's mammals or birds or insects, to do their work for them. This is another thing that's like noteworthy in terms of speciation. We see gymnosperms speciated because they moved into parts of the land that were previously unoccupied by plants. Angiosperms are going to speciate because all of this coevolution that takes place with their flowers and their specific pollinators. Fruits are not always fleshy. They're not always full of sugary nutrients that are like an apple that are going to pay off an animal for delivering it great distances. what makes a fruit a true fruit, like we said, is it's this wall this ovary wall that surrounds a seed. So like we said, tomatoes, that's a fleshy ovary wall that surrounds a bunch of seeds. That would be a fruit. These peppers would also be considered fruits rather than vegetables. Something that would be a true vegetable doesn't have seeds in the middle of it. So think of like a potato or a carrot, something like that. This is an example of a Buckeye plant. It's an example of a fruit that doesn't have a fleshy wall. So this would be a fruit that is not paying off some animal to disperse the seeds. This is one of the examples of a fruit that does mechanical dispersal. These Buckeye plants, they form this, it's a hard pod that dries out. And when it dries to a certain threshold, it's like it hits an eject button and it pops open like this. And what comes out of it are these seeds. They look like the eye of a deer, so they're called Buckeyes. But that popping open of this pod throws those seeds out just a short distance from the plant that produces them. Let me try this again. All fruits are going to aid in the dispersal of their seeds. Some fruits are going to aid in the dispersal of their seeds mechanically by ejecting them out into the environment. There are more common ways of seed dispersal, like water is going to help the seeds disperse. We'll see wind as well as animal dispersal. This is a dandelion picture. Dandelion seeds are right here, and they're attached to those little wispy structures that carry them by the wind. So dandelions would be an example of wind dispersal. If we're thinking about water dispersal, coconuts would be an example of water dispersal. Mangroves, like red and black mangrove trees, those are also dispersed through the water and can land on islands. So water dispersal happens, but maybe the most common method of seed dispersal is going to be employing the help of some type of animal to disperse those seeds to a new location. I want to show that in a video real quick, but the last few slides that we go through in our Angiosperm PowerPoint are what distinguish the two groups of angiosperms that we're going to need to see. This is really setting us up for next week. We're going to end up taking a tour of campus plants. We're going to find maybe 10 to 15 different campus plants. And one of the things we're going to ask about those campus plants, what group does it belong to? If it belongs to an angiosperm, we're going to need to know whether it's a monocot or a dicot. So let's talk about 4 different characteristics between monocots and dicots. You'll also notice that I'm up here saying dicot, and the screen has put the word "u" in front of dicots. That word "u," that prefix just means true. So eudicots, that's the same thing as what I've written on the board here, that I'm just saying dicots. I'll use the shortened version of that, but it means the same thing. So when we compare monocots and dicots, the first characteristic that we can see that's different between the two is that monocots are going to have parallel veins, where dicots are going to have branched veins. What does that mean? Here's what it looks like. If you grab a leaf, and we will have plenty of these in here next week. We have one of them over there right now that we can grab. It's our only angiosperm. If you look at these leaves, you're going to see veins that are branching out. They're like the one on the left, where they have this network of veins that branch away from this mid vein. If it branches, it's a dicot. If the veins run parallel to one another, like you would see in a grass, all of the grasses are monocots. Something like bamboo, it has parallel veins. It is also a monocot. So parallel veins, monocot, branched veins, it's a dicot. That's 1. 2 on the list is the type of root. Sometimes you might not have leaves to go off of. You might only have root to look at. There's a difference between dicot roots and monocot roots. In a dicot root, there is a central, kind of a main root that we call a taproot. And then you can see off of that main taproot, there's a bunch of smaller roots that all branch back to that main taproot. So if we see a taproot system, we know right off the bat that it's a dicot. In the monocots, there is no main taproot. It's just a bunch of collections of individual roots. This is what's called the fibrous root system. There's no main taproot. Monocots all have a fibrous root system. Dicots are all going to have a taproot. That's 2. The third characteristic has to do with if we are able to see these plants right when they germinate, right when they pop out of their seed coat. We said that what's about to pop out of that seed coat is a baby plant. It already has some starter roots and it has these little starter leaves on it. The term for those starter leaves are called cotyledons. That's not a very common word, cotyledon, but it's where these two groups get their name. Monocot or Dicot. The cot ending comes from this term cotyledon that refers to embryonic seed leaves. It has to do with how many embryonic seed leaves do you have when you, as a growing plant that just pops out of this seed coat. Some plants have 2 embryonic seed leaves. Those are the dicots. And other plants, like all of the monocots, this is a corn seed that is growing. Corn is got parallel veins. It's got a fibrous root system. It also has 1 cotyledon. So 1 cotyledon is where these get their name monocot. If there are 2 of these embryonic seed leaves, then we know we're looking at a dicot. Embryonic seed leaves are not present all the time, so it's just one of these characteristics that we use to distinguish monocots from dicots. The fourth and final characteristic, one more slide. This is what we just saw as the seed. We can see there's that little starter root. And then there's the tip of the shoot. You can see on either side of the tip of that shoot, there is an embryonic seed leaf that is just waiting to pop out of this seed coat. So before this thing even pops out, we can tell we're looking at a dicot. It has 2 of those cotyledons in there. So again, cotyledons is our third characteristic. If it has 2, it's a dicot. If it has 1, it's a monocot. Now for our fourth and final characteristic that distinguishes monocots from dicots. If we have a chance to find flowers and we can look at the floral parts, if it's a monocot, the floral parts are going to be in multiples of 3. Let's look at this grass as an example. The grass, there's the flower of this grass, and we can see male stamens, and there's only 3 of those male stamens. So if floral parts are in multiples of 3, it's a monocot. Here is an orchid. We've got 3 of these pretty showy petals. Those little splotchy looking structures are the sepals. So we can see 3 sepals, we can see 3 petals. Multiples of 3 tell us that we're looking at a monocot. If the petals had multiples of either 4 or 5, then we're looking at a dicot. So there's petals that has multiples of 5. If I go back to those slides, there were some flowers we saw earlier. What were those? Here's one. It's got petals in multiples of 4. So we know that would be a dicot. This one, what do we got here? We can't see all of the petals, but we can count those anthers. There's 1, 2, 3, 4, 5. How's that? 1, 2, 3, 4. There should only be 6 there. I'm not sure. That might not be. That might just be kind of an optical illusion on one of those. 1, 2, 3. Should be 6. This should be a monocot. That is not clear enough to be a test question. But this one would be, we can count petals, 1, 2, 3 petals, and we can even count those anthers. There's 1, 2, 3, 4, 5, 6 of those anthers right there. So floral parts in multiples of 3 tell us we're looking at a monocot. Okay, back to these things. What about this one? 1, 2, 3, 4, 5. That tells us we're looking at a dicot. I don't know if we can see all the flowers on that one. I'll just skip over it. Okay, monocot versus dicot. We got all the way through those, and I think that might have been the last slide. So the last handout that I'm going to give you is the one that shows angiosperms in action. Let me know if I missed any of the explanations from the conifer handout. I'll be glad to fill in any of those if I did.